Okay, so basically hereditary and variation. So we're going to be focusing on this particular topic, and basically when it comes to hereditary and variation, it's basically talking about how genes are passed on to another generation, to the next generation, and so on and so forth, right? And obviously that's hereditary, right? So how genes are passed on, and then variation really just talks about genetic diversity and basically how species can be different as they move from generation to generation, what changes they make, and how does this variation basically benefit them. So basically, hereditary and variation is a part of both the syllabuses in the CSEC biology syllabus, and it's also a part of the CSEC HSV syllabus, right? So that's the plan. So it's kind of going to be like a quick short course, not short course, like a crash course, and basically hereditary and variation. So just going over the main pin points that we need to mention if we're going to be talking about hereditary and variation. So what's the plan? So the plan is hereditary, we're talking about DNA, we're talking about genes, we're talking about alleles, chromosomes, and different types of genes. We're going to be talking about cell division and obviously variation, and then monohybrid, dihybrid crosses, and then complete, incomplete, and codominant. So that is the plan with all of this stuff. So, as we said before, hereditary. So basically, we're talking about how genes are passed on from parents to offspring, right? So, for example, let's just say my parents, or let's just say, yeah, my parents have brown hair, right? One parent have brown hair, and then one parent have black hair, right? And then I am the offspring, right? So hereditary is all about how those genes are passed on. So one of my parents have brown hair and the other one have black hair. So what color hair do you think I'm going to have when we mix? Because this is what we're talking about. This is a little bit of genetics, right? So we're talking about genetics when we talk about hereditary and variation, right? So we have to talk about that. So basically, what hereditary and variation and genetics, all of those, they're trying to explain how the offspring end up the way it end up basically. So the question is, if one parent have black hair and one parent have brown hair, which color hair the offspring is going to have? So basically, that's what hereditary explains. So basically, it's the passing on of genes from parents to offspring. And then variation talks about the differences in those traits between individuals or groups. So as I said before, variation is really and truly talking about the differences, right? So for example, because okay, one way to remember it, when you say the word variation, think different, think genetic diversity, because that's basically what variation is talking about, right? So for example again, and we can, as we go along, we're going to explain these things a little bit better, or we're going to learn the biological concepts that can explain these things a lot better. So for example, if we're talking about variation, let's just say maybe diabetes or something running at the family, and let's just say the next generation come and they don't have diabetes. So you're probably say, that's different, right? And basically this difference in the genes or genetic makeup is what makes these offsprings, the next generation, a lot more stronger. It makes them less susceptible to diseases. It just overall helps them to live longer because they're different and they're able to adapt to the environment differently, right? So that's the main difference between hereditary and variation. But overall, these two concepts are what helps us to understand how traits are passed on from the parents to the offsprings. Right, so now if we're going to be talking about anything hereditary, if we're going to be talking about variation, if we're talking about anything genetics, we have to talk about DNA, because this molecule right here is the backbone. It is the backbone, it is the foot, it is the everything. It starts from here, right? It starts from here. Now, basically, every living organism has a DNA. So it's either they have DNA or they have RNA, but RNA is only in viruses, right? But we're not talking about that right now. What we're talking about is specifically DNA. So it's deoxyribonucleic acid, right? This molecule, deoxyribonucleic acid, carries the genetic information, right? It carries the genetic instructions for basically our entire being, right? It is what we make up of. So for example, if you ever hear like a person saying, oh, let's just say that a couple, right, or they were a couple, then break up, and then the male say he want a DNA test, right, for the child because maybe you don't believe the mother or something. But let's just say the father want a DNA test. And that is why DNA is so important, because why you think him want a DNA test? Why him test the DNA? Because that is where the genetic instructions are, right? And if a person, and within families, you share DNA, right? You share some form of DNA with your parents, with your cousins, with, for example, your cousins, people like that. It gets lesser and lesser, right? But for somebody as close as like a father or a mother, you will find a good amount of the same genetic makeup, right? The DNA. So basically, it carries the genetic instructions for development, functioning, growth, and basically reproduction. Because let's just say that one of my parents them are really short, right? And then me come out and be really, really short. Why me come out really, really short? Because one of my parents were short, and it's because that gene got expressed in me, right? So we're sharing the same DNA, and that is why I look the way I look. That is why you probably look like your mother, you look like your father, you look like somebody, but you look like somebody in your family. It's all about the DNA, the deoxyribonucleic acid, right? That genetic molecule that codes for everything down the line, right? So without DNA, you are not you, right? It starts from here. Now, if you look at this diagram, so the structure of DNA is known to be a double helix, right? So it's a double helix, it's two strands, right? It's a two-stranded molecule. That's how we say a double helix. So that's how the structure of DNA would basically look. Now, if we're talking about genes, now genes are also very, 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 very important, because genes are basically a segment of DNA. So if you could look at this diagram right here. So before we say that this is a double helix, this is the DNA molecule that is within us, right? It is what because this is me. We need DNA for anything, for like cell division, right? The fact that we grow, we need it all starts from DNA. And maybe if you remember the process of cell division, if you remember mitosis or you remember meiosis, you would, and we're going to talk about that later on, but you would remember and know that within the nucleus, that is where you find the chromosome, right? Right, and inside the chromosome you obviously have the DNA, and then DNA you're gonna find a gene. So the gene is basically a segment of the DNA molecule. So if this, for example purposes only, if this was the entire DNA molecule that we had inside of us, that small section, that small segment would be the gene, right? And it's basically a fundamental unit of hereditary. That's what the gene is, right? So it contains instructions that are specific to make the specific protein or the specific RNA molecule, but RNA is mainly come up in viruses, but obviously it plays a role in what do you call it now? We can't remember right now, but RNA is also important in our DNA replication process, yes. So that's some molecular biology business, but we're not getting to that. But yeah, so genes help to dictate an organism's traits at the end of the day. So if you look at this is what I was saying. We have the nucleus. This is the cell nucleus. You have all these chromosomes inside of it. And on these chromosomes, you're going to find a DNA molecule. You're going to find DNA. And where you find a DNA, you're going to find the genes, right? So all of this come together in order to make us us, right? And remember, in our bodies were made up of millions, millions, and millions of cells, because cells are really, 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 really small. This diagram is showing an animal cell. Look, can you imagine that? We're talking about DNA, and we're talking about genes, right? And look at this diagram. A cell can only be seen under a microscope, and then you're telling me that I'm telling you that after you see this cell under the microscope, it gets even more complex, because you have chromosomes in that nucleus, right? You have chromosomes in it, and you also have your DNA in it, and you have the gene, which is a segment of the DNA in it. So it's a really, really complex process that sometimes you probably can't wrap your head around, the fact that, okay, the cell is already so small and so much is going on inside of it, right? But just remember that the gene is a fundamental unit of hereditary. So, and it's a segment of DNA. So once you see the DNA molecule, which is a double helix, just remember that a segment of that is a gene, and it codes for a specific trait, maybe blue eye color, maybe black hair, but it codes for something. Right, so the same chromosome that we were talking about, that is inside of the nucleus, right? And these chromosomes are basically the stars during cell division. So during mitosis or meiosis, they line up at the equator, they become condensed, they start out as chromatids and all that stuff, but they're basically show behind cell division. But just to get an idea of what your structure is like, so it's like a thread like structure, right? So basically this representation is when they are condensed, right? So when they are decondensed or uncondensed, it makes them a lot harder to see. It makes it a lot harder for us to see, right, when they are not condensed. So when they're condensed, they resemble this, but in all reality, they are made up of thread-like structures, right? And if we get a little bit more complex, like some nucleosomes and stuff like that, because it's literally a thread-like structure, and we can reel it all out. We can literally reel it. So this chromosome, if we take piece, I would just start is literally strands of nucleosomes packed together that make up the chromosome, right? But that's not the point. We just want to know at this level that it's a thread-like structure within the nucleus, and it carries genetic information in the form of DNA, because we know that. And by the way, DNA are also packaged with proteins and all that stuff in here, right? But we're not getting into any of that right now. But basically, the chromosome, and we know that the DNA can be found on the chromosome, and then we know that the gene can also be found on the chromosome, right? We know that. So that is why it carries the genetic information, because it carries all the structures that code for our genes. We have our gene, we have our DNA, everything is locked within these chromosomes, right? So now if we get, so bear in mind, or let me just repeat again, that a gene is a segment of DNA. We said that before. Now we have different types of genes. So basically we can have a dominant gene, and we can also have a recessive gene. So a dominant gene would be a gene that is expressed, meaning that it shows up, right? And then the recessive gene would be one that is not that is present but not expressed, right? So we say that dominant gene is represented by a capital letter. In this case, you're going to use capital B. And then recessive gene is represented by a common letter, and in this case we're going to use a common B. Now, how I would explain this is that, as I said before, we said that genes code for a particular trait. So now these B, and this is example purposes only, because if we want to represent the gene, we can use anything. We can use a big A, a big B, a big C. It depends on what trait we're coding for, right? So in this situation, this big B is going to represent black hair. Me, I'm gonna make it represent black hair, right? But if you get a question or something like that, you might see C, you might see big R for red flowers. It depends on little R for white flowers. It depends on the question, it depends on the gene and all that stuff. But in this example, just know that the dominant gene is always the capital letter and the recessive gene is always the smaller, the common letter. But in this case, we're going to let capital B code for black hair, right? So this is going to be black hair, and then the recessive gene right here, the recessive gene, we're going to let the recessive gene code for brown hair. So it means that in this situation, and bear in mind that you get two, right? You get one from each parent. So it means the example I'm used before, say one of my parents have black hair and one have brown hair. But in this situation, brown hair is the recessive gene, right? So let's just say that my father did have black hair and me get this gene from him, capital B, and let's just say that my mother have brown hair and me get this recessive B from her, and because it's recessive, so basically these two B's, the capital B and the common B, both of them make up who I am right now. That is the gene that me end up with, right? Because we get one from each parent, right? And if we go back to sexual reproduction and we go back to fertilization, we know that it's both parents involved, so the offspring is going to be determined by the genes that them get from both parents, right? If it was asexual reproduction, there's just one parent, so whatever the parent have, that you get back. There's no question or none in it. But since it is sexual reproduction and we know that we're going to get one gene from each parent, and I said before, if my father have black hair and my mother have brown hair, but the gene that she passed on to me was recessive, right? And the black hair gene, the black hair trait which my father gave me is dominant. So when they made me, and this is me now, and me end up with a capital B and a common B, what do you think would be my hair color now, right? So this codes for black hair, this codes for brown hair. Me get both of them. What color hair do you think I'm going to have if my parents give me both these two genes? My hair color is going to be black, right? And it's going to be black because it's the dominant gene. It means it overpowered the little b. The little b is not going to be expressed. It calls for brown hair, so it means that, let's just say in the future, if me meet somebody, if me have some kids, one of my kids then probably will end up with brown hair, right? But at this stage and how they made me have black hair because that is a dominant gene. The black hair, dominant hair, the brown hair is very much recessive, right? But one thing to note is that a recessive gene, when you have two recessive genes, it will be expressed. So for example, if me never have a capital B from my father in this example, and we have if there were two common B's here, that may get one common B from one parent, one common B from the other parent, so brown hair come together, obviously we're going up with brown hair, right? Because the only time a recessive gene is expressed is when both of them come together beside each other. That's the only time, right? That is the only time it's going to be expressed. And the next thing to note is that with the recessive gene, it can show up later on. So you might pass this generation and next generation, and you just say you just don't see anybody with the brown hair, right? But it does not mean that the person is not a carrier, and it does not mean that the person does not have the gene. It just means that their gene is recessive. That means it's like dormant. It did it, but it cannot be expressed. And so if you keep on if an entire family keep on reproducing, meet people and reproduce new offspring, and let's just say that a pair dominant, right? Pair dominant gene the child gets, then the brown hair does not go expressed, right? But it does not mean that the gene is dead. If one person in the family buck up on somebody who have the recessive brown hair, and then brown hair and brown hair come together, the child again up with a brown hair. So that is why in some situations certain things can skip much generations and generations and generations, and then 10 generations later, something that you think they did out long time a show up back, your great grandkids or something like that, right? So that's basically how the dominant and the recessive gene works. Okay, now then you have so if we're talking about genes, right, they can also be homozygous and they can also be heterozygous. So we said before that the recessive one is not the recessive gene is not expressed unless there are two of them together, right? So if a big B is before the little b, that little b is not going to be expressed, right? So homozygous, which means same, means that two alleles or two genes, basically it's just basically it just means that both of them are the same. So in the example here we have 2b, so you have big B, that's homozygous because both alleles are the same, both genes are the same, right? Big B. And then you have heterozygous, which means that different. So it means that one of them are different. So you have big B, little b. And obviously recessive, right? You have little b, but in this situation, if we put another little b right here, if we put another little b, if we put another little b right here, that would be homozygous recessive, right? So we can just say homozygous recessive. Right, so that could be homozygous recessive. So you have homozygous dominant, you have heterozygous, and then you have homozygous recessive. Those would be the three, right? Those would be the three main ones. Now, if we're comparing the genotype now versus the phenotype, so basically when we talk about genotype, we're talking about, and an easy way to remember this is the word geno. Just remember genetic because both of them start with G. So the genotype is really talking about an organism's genetic makeup, and the phenotype is talking about the physical characteristics that me and you and everybody can see. So genotype, genetics, and then phenotype, physical characteristics, right? So in this situation, if we had a genotype, we have big B, and we say that B, the big B equals our black hair. So this is the genotype, right? This is the genetic aspect of, yeah, big B, and then the phenotype would be the black hair color. Now, so basically, alleles, they're basically a form of a gene, right? So there are a pair of genes that occupy a specific location on a particular chromosome and they control the same trait. So alleles, they are a form of a gene, and examples would be hair color, eye color, flower color. So they're just like different versions of a gene, right? So that is what alleles are. Then we want to find out why is variation important, right? So we said before that all of this, the entire point of all of this is to really see how genes are passed on from one generation to the next. But how does something like variation drop into this, right? How does variation drop into this? So basically we talked about that variation, we're really talking about what is different between the generations or what is different between the species, right? And so basically variation is extremely important. Why? Because it leads to genetic diversity, right? It's necessary for survival of any living organism, and basically it helps organisms to adapt to new environments. It builds up like resilience so that a species does not become extinct, and it also makes organisms less susceptible to diseases, right? So imagine, and by the way, the variation would come from sexual reproduction and only sexual reproduction. Something like asexual reproduction, if we have a flower, it produces asexually, right? And it just create a bunch of offspring just like that. Let's just say that the flower is susceptible to a particular disease, right? And then it will have like 20 offsprings that exactly like it. What's going to happen is that if that disease that can basically kill the parent, I will kill off the entire 20, right? And that puts the species at risk for extinction because them can't adapt to the environment. Them have the same exact trait, them have the same exact genes, them can't adapt to the environment. So it means that the parent are dead and all of the offsprings are going to die as well. What mean extinction, right? So that is why variation is important, because even for human beings, if you look at human beings in the sense that what if all of us were exactly the same? For example, like a flu come, you know that some people can actually die from flu, right? Because if the immune system really weak, they can die. So like for even people with HIV and stuff like that, and the virus weakens your immune system. So for example, if me catch a little flu, we can get over it. And let's just say somebody with HIV, them catch a little flu, and because we know that their immune system is very weak because the virus take over. Basically, if they get a little flu, it's not a little flu for them because it will affect them body way more than it would affect me with quote unquote normal, right? So what's going to happen is that yes, so let's just say it affect them body really bad. So it makes them more likely to die because you might say, oh, let me just die from a little flu. But listen, if their immune system is really if the immune system really down and them catch a flu, I eat that for them because their body cannot fight back. The T cells them cannot fight back. They cannot respond, right? Because the virus hijack themselves. So what if that person with HIV, what if everybody was like that person and basically everybody have the HIV. It means that just like how that person could die from it, it means all of you could die from it too. But in this situation, we're talking about variation and we're talking about diversity. So it means that different people are different, right? Different organisms are different, different species are different, right? And it's necessary for survival because the truth is that if a disease come right now, some of you are dead. Some of we go dead, some of we go live, and some of we depend on the borderline, right? But it's very important to have variation. It's necessary for survival. And the HIV example, we only use that example because it comes to my head off the bat, but this is not anything related, this is not anything stigma related. We just want to make that clear. But at the same time, HIV does weaken the person's immune system and right? So it's not necessarily the HIV that kill the person that can kill somebody, but it's just the what it does to the body, meaning that it hijacks the body's cell. It's a virus, and it weakens the immune system significantly, where T cells cannot fight back. So that is why variation is important. Or somebody with asthma, basically people have different illnesses, right? Variation, and for example, maybe as I said before, maybe diabetes run in the family, but meaning you're more likely to get it. Not that you can have kids and it pass on just like that, but you're more likely to get it if it already run in the family. So it just means that you have to be more careful with your diet and stuff like that. But even in that situation, the fact that just because your grandmother did have it, that means your mother got diabetes. Just because maybe if your mother have diabetes, it don't mean that you must necessarily get it, right? So that's really what variation is and what it's basically talking about, right? And we said before that if we're talking about variation, it will come from sex reproduction, the fusion of male and female gametes, some fertilization have to take place, right? And another way that variation can come about is through a mutation, right? So mutation meaning that there's a change in the DNA sequence, right? So for example, during maybe cell division, we know that during cell division, during the S phase, that's where DNA replication takes place. Let's just say something went wrong, the spindle never attached properly, something go wrong, a mutation happened, right? And then bam, you realize say you're gonna be different, or I don't know, something gonna change though because it never um line up the way it should, or it never attached the way it should. So that's basically like a what's the word? It's more like you're more likely to have an abnormality, yeah, something like that, abnormality. And another way that you can get like a mutation happening, right, is through mutagens such as radiation. That is why them tell us to stop microwave food, cook food, stop microwave food, stop be close to like radiation, heat, anything like that, because it's not good for us. So that's why they say that. And not all variation is good, as you can see based on the whole radiation thing, because you know that can kill you, and radiation can trigger the cancer cells and bomb you have cancer, different things. So it's not all variation is good, but it can basically change things, just know that. Okay, so now cell division, mitosis and meiosis. So we're going to focus a little bit on cell division, and Not going completely in detail about it, but we're just gonna run through cell division quick and fast. But basically, there's a more in-depth, I think we call it in-depth video about cell division, so you can definitely go and look at that, both meiosis and mitosis, so you can look at that. But now we're just gonna run through really quickly, right? So as it relates to the stages, well, okay, so we're not going to go through the stages of interphase. Yeah, that's what I mean to say. But we're going to focus on just PMAT. So basically, cell division is the process by which new cells are formed, and the main two types of cell division are mitosis and meiosis, right? Now, there's a difference between mitosis and meiosis, so it's not just one cell division take place in our body for everywhere, right? No, there are two main types. We have mitosis and we have meiosis. Mitosis, that's where our body cells slash somatic cells, they divide. So the word somatic is really talking about body cells, so same thing, body, somatic, same thing. So in mitosis, division of body cells or somatic cells to produce two diploid daughter cells. So we know that the end result of mitosis is going to be 2, and it's represented by 2n. Meiosis, on the other hand, is a specialized type of cell division, right? Because it only focuses on our sex cells, right? So this is how we basically reproduce to have like actual offsprings. No, mitosis is really for like bodily repairs, maybe I get a cut, cell division, fit, heal, and stuff like that, right? So repair and yeah, just mainly repair and build up cells, maybe um replace worn out cells. So it's really repair and replace of worn out cells in the body. That's what mitosis is. While meiosis specifically, we're talking about offsprings in this situation. And the end goal of meiosis is going to be 4 haploid daughter cells, and the haploid is represented by a N. So this is the process of mitosis. So basically, how I remembered it is PMAT. So prophase, metaphase, anaphase, telophase, and then you have cytokinesis. Now, I said PMAT. So why we include cytokinesis? We're going to explain that. But even before we talk about these stages, we have to talk about what happens. We're not going into too much details, but we're just going to brush over it. Interphase. So interphase is that preparatory stage that the cell is basically in before it starts to divide. Now, the cell spends a lot of time, in fact, spend way longer in interphase than it actually spends in the actual mitosis process, right? And interphase comes before both mitosis and meiosis because that's where it's like a resting stage. The cell is just in there growing. It's just getting ready to divide. No, I'm never gonna mention the stages, but we just brush over them. So in basically in interphase, we're going to have DNA replication taking place. We're gonna have the cell growing and then growing some more. That's really interphase, right? So the cell spends a lot of time in that stage. Now let's start focusing on mitosis. So after interphase done and gone and the cell enters mitosis, what's going to happen is we're going to start with prophase. So what's happening in prophase is that these chromosomes are going to become condensed. So as you can see right here, these two are the chromosomes in this situation, right? And this example only shows 2, right? But there can be a lot in there. But this example only shows 2. But this is where the cell is going to, and the chromosomes become condensed, and then you'd also see the central, what's it called, the centrioles. So the centriole or the central, let's just say centrosome, because the centrosome has in the centrioles, the centrosome, which is basically going to be like two, like a circle or something. They're going to be on opposite ends of the cell. This diagram don't really show it, but it's going to be centrosome. It's going to be an opposite end of the cell. Centrosome is going to be there. Is going to separate the opposite ends of the cell. That's going to happen, and the chromosomes become condensed. So that's really what's happening in prophase for the most part, right? And the reason why they're becoming condensed is because before they were not condensed, they were uncondensed, they were decondensed, they were not showing, right? And now they're showing, so they're condensed. So that's prophase. So as we move into metaphase right now, by the way, in the middle of these chromosomes, so basically one of these are called a chromatid. So these are two chromatids joined together by that circle ball. The circle ball is called a centromere. So the centromere is holding the two sister chromatids. So individually they are chromatid. One of them is a chromatid. Two of them is a chromosome. So when they're joined together by that centromere, the little ball in the middle, it becomes a chromosome, right? So back to metaphase. In metaphase, we said before that we had centrosomes at the opposite ends of the cell. So this diagram not sure, but it would be like a little circle ball, right? Circle ball. And what's happening in this metaphase is that the chromosomes become lined up at the equator. So this right here is middle equator. It lines up right here, and from these centrosomes or centrioles, we're going to have these spindle fibers extending. When these spindle fibers extend, they are going to basically attach themselves to something called a kinetic core. So the kinetic core is at the back of the centromere, so it's going to attach to the kinetic core, which is on the centromere right here. So these spindle fibers, they extend, they attach themselves, and we have the chromosomes lining up at the equator. Those are the main stuff that are happening in metaphase. As we move down to anaphase, what's happening here is that, and how you remember, by the way, just a way to remember, prophase, pro means before. So before, as you can see them just in there and I really don't much, and it's become condensed. And then metaphase is from a middle, they line up in the middle. Anaphase means away. So in this stage, these chromosomes are being detached, right? So when you have like a force, two opposite forces, it's going to split it apart. So in this case, what's happening is that the sister chromatids are split apart again, and they're pulled to opposite ends of the cell. That's what's happening here, right? Because two of them were chromosomes, and now they're pulled apart, going to opposite ends of the cell in anaphase, right? So they're pulled away from the equator. Now, what's happening in telophase is just that after they've gotten to opposite ends of the cell, we're going to have a like a membrane forming around them, right? Actually, yeah. So basically, they have like its own individual like nucleus forming around it. You know, when if that would be the right word, well, let me just work with it right now. But they can, yeah. So they go to opposite ends of the cell, and as you can see, yeah, I don't want to mention that, but yeah. So they're pulled towards the end of the cell, and you have equal amounts on both sides in telophase. And then cytokinesis is what is going to split them into two. So these two are going to become the daughter cells. Two of them will become daughter cells. So the goal behind the entire mitosis is to produce two daughter cells at the end of the day. And as you can see, we end up with equal amounts in each daughter cell. So that is the goal of it. And also in telophase, this is where the we call it the nuclear membrane. So the nuclear membrane starts building up. So that's what's happening here. The circle around it, the membrane is the nuclear membrane around it, right? So it's a nuclear membrane, and the chromosomes start becoming decondensed. As I said, this diagram is not the best diagram, but yeah, they become decondensed. So you see how visible they were? That means that they start not being as visible, right? So the end goal is kind of like for data cells to look similar to what we started with initially. But what's happening? Nuclear envelope or nuclear membrane rebuilds, chromosomes become uncondensed in telophase, and then at the end we end up with two separate daughter cells. That's basically mitosis. Then in meiosis, it's basically way more complex. It's complex, but at the same time, it's the same thing. Okay, so what's happening is here in the prophase. So what's happening is we have PMAT twice, right? We have PMAT twice. So it means that the cell is going to have a prophase 1 and a prophase 2, a metaphase 1, a metaphase 2, and a phase 1 and a phase 2 is going to have a telophase 1 and a telophase 2, right? Because at the end of the day, we're going to end up with 4. That's the goal, right? The goal is to end up with 4. So in mitosis, we ended up with 2. Meiosis, so we basically do mitosis twice. This is, we can just kind of look at it like that. And yeah, we do it twice. So we're gonna have double the daughter cells at the end of the day. So first in prophase, and basically the first round of meiosis is basically just mitosis, right? So you're going to have, but bear in mind that meiosis is the division of sex cells, right? So we're talking about genetic variation and all of that, and we get one from each parent, right? So that's the difference with mitosis and meiosis. So prophase 1, the nuclear envelope is going to break down, chromosomes are going to be condensed, and then something that did not happen in mitosis, which is very important, is crossing over. So crossing over is basically where these diagram don't really show it and so good, but basically if you look at this one where you see the red have the blue on it, this one with the red have a blue on it right there, that would be crossing over where piece of like a segment off of one chromosome cross over and go on the other one, right? So that's how we get the genetic variation. So the nuclear envelope is gonna break down, chromosomes become condensed, crossing over takes place in prophase 1. Metaphase 1, same thing, line up at the equator, line up in the middle, and then we have the centrosome, what I talked about earlier, and the centrioles, right? So in metaphase, the centrosome is going to they're going to extend the spindle fibers, chromosomes are going to line up at the equator, right? Now there's something that is very important here. So in metaphase 1, you can see them line up at the equator, and then if we just jump ahead a little bit and look at metaphase 2, they're also lined up at the equator, but what's the difference is that they're lined up in different directions. As you can see, you see it? They're more horizontal over here, so them very vertical. They line up differently. And also, the next thing we need to make mention of is that we don't just have two chromatids joined together by a centromere forming one chromosome. What we have going on is we have homologous chromosomes in the nucleus. That's what we have going on here. We have homologous chromosomes, and these homologous chromosomes can only be found in meiosis. You cannot find them in mitosis. And as we said before, because we're getting from both parents, right? So that is why we get chromosomes from both parents, right? So at the end of the day, we have 43 chromosomes in total. Also, right? No, we have 46. Sorry. We have 46 chromosomes in total, 46, 46, and we have 23 chromosomes from each parent, right? 23 from each parent, 23 and 23 make 46. So we have these homologous chromosomes, they become condensed, nuclear envelope breakdown, crossing over take place. That's how we get the one version. That's how we get one way of getting genetic variation. I'm soon tell you what the other one be. And it's within these steps. So the metaphase 2, they line up at the equator, spindle fibers extend, bump to the joint to the kinetic cores at the back of kinetic cores on the centromere, right? So spindle fibers extend, attach themselves, anaphase the chromosome. So before homologous, or we could also call them a tetrad. So that's basically 4 chromatids, 2 chromosomes, 4 chromatids make up the homologous pair, right? So here when we have pulling apart is we have one chromosome and so, one chromosome and so, right? And you can see the genetic variation. You can see what happened because of crossing over, that we have different color and different color, right? So that's what happened. Then in anaphase, you're being pulled opposite ends of the cell, And then in telophase and cytokinesis, the nuclear envelope is going to reform, chromosomes become decondensed, meaning they're going to be less visible now, and obviously the spinal fiber is gone, and it's going to split into, it's going to create a little hunch in it, like kind of like split into two, right? So now prophase 2, right? But bear in mind, so even though it's splitting into two, it don't split come, what is it? It don't really necessarily split completely, right? So it now got the pinch off right here, so not here, so not time it. But in prophase 2, it's basically the same thing that gonna happen. Chromosomes will become condensed, and you know the centromeres are going separate the opposite ends of the cell. Metaphase 2, these are chromosomes lining up at the equator. In metaphase 1, we had homologous pairs, we had tetrads lining up at the equator, right? We had 4 chromatids, 2 chromosomes. Here we have 2 chromatids and 1 chromosome, right? Lining up at the equator, and as you can see, in metaphase 2, they line up more vertically. Anaphase 2, the chromosomes are both opposite ends of the cell, right? And then we're going to have telophase and cytokinesis, where basically nuclear envelope is going to break down, become decondensed, meaning less visible, and then you're going to split. So at the end of the day, when the cell finally does cytokinesis and splits, we're going to end up with 4 daughter cells, right? So cleavage barrow is right here, but it now completely split. So what's going to happen is this would enter in over here. So I would run it again, I will end up with 4. The end of this diagram don't necessarily show the 4, but you get the point. Then the last thing we're gonna mention is really about well, this is not the last thing for about other thing, but yeah, different inheritance. So basically how inheritance works. So we said the entire point of all of this is to find out how genes are passed on. So what's happening here, we're going to talk about the monohybrid cross first, and then we'll talk about dihybrid afterwards. So in a monohybrid cross, it's a genetic cross, right? So this is how we can kind of like predict the next generation, right? So it's a genetic cross between 2 homozygous individuals, right? Remember what we said homozygous meant, they must have the same thing. Now in this diagram, this will be the F1 generation, meaning the first generation. So if the parent had a child, this would be the first generation afterwards, right? The offspring is the first generation. We said homozygous. You can have homozygous dominant and also have homozygous recessive. So if you cross a homozygous dominant with a homozygous recessive, so if the one parent had black hair, so BB, which goes for black hair, which, well, not black, oh no. So if BB, forget, so we do a different example. Let's say we do flowers, my bad. So we're doing flowers in this example. So BB, so the big B is blue, which is dominant, and then the little b is white, which is recessive, right? So if we coded big B, which goes for blue, which is dominant, and then we code homozygous recessive, which goes for white, what's going to happen is that we're going to get 100% 100% blue flowers, right? 100% blue flowers, right? So as you can see before, this is the genotype. So it means that all of these flowers have the like the trait for white flowers, but it's not expressed because the dominant one overpowers it. So it's 100% blue flowers that I get, and then I get 100% for the genotype, you will get 100% big B, little b, and then for the phenotype, 100% blue flowers. So where things gonna get more differentiated with our monohybrid crosses when we go down to the F2 generation, and that's basically the second generation. Now we're using a different example. So what's happening here is we have 2 homozygous, well, not homo, 2 heterozygous individuals, yeah. So let's just say right here we have all of these individuals are heterozygous, by the way, right? Because they have 2 different genes, two different alleles, right? So yeah, so usually at the end of the F1 generation, you're gonna get heterozygous. Now when we go to the second generation, right? Meaning heterozygous. So if an heterozygous crosses with another heterozygous, which in this case we're going to be doing big R for red flowers, and then big R, little r for also red flowers, and then little r for white flowers. So red flowers is the dominant trait, and both these genotypes code for it, right? And then little r, that's white flower, which is recessive. So if we cross two heterozygous, meaning big R, little r. So that's the genotype, big R, little r, and big R, little r. So that means that we're crossing two red flowers, right? So it's two parents and both of them look red. What do you think could happen? But they have different genes, they have different alleles, and we say that alleles are basically different are basically alternative forms of a gene, right? So it can code for different things. So in this situation now, we have two heterozygous, both of these flowers look red, but if we want to predict the next generation, we can do a cross, and that's what we're going to do now. So the first child them have, and this is just a Punnett square that we basically use to do it. It's called a Punnett square. It's very easy. It don't hard. Just draw four box, and then you get the alleles of big R, little r, big R, little r. Because in this situation, it is heterozygous crossing with an heterozygous. We know that both heterozygous they code for red flowers. So now two flowers that look very red, we're going to cross them, right? And then the first child that I'm have, so this first 25% is going to be red flowers. So it knows, okay, the first child will come out completely red flowers, right? And them not gonna have no trait, them not gonna have the trait for white flowers, because them not like a R. So it means say them fully red, them not no trait for white flowers. Then the other two, which is 50%, we realize they both have the white flower trait that carry with them. So that's 50% for the genotype big R, little r, and the phenotype is going to be red flowers, right? And then the last one is going to be little r, which is recessive, and it's going to go for white flowers. So at the end of the day, we have one, two, three. We have three red flowers, right? We have three red flowers, and we have one white flower, right? So 3 to 1, right? And then for the genotype is going to be one, right? One big R, two big R, little r, and then one little r, right? Right. So this should actually be 3. So it's not a mistake. It should actually be 3 to 1. So the phenotypic ratio is, we say that the phenotypic ratio is the physical characteristics. In this case, it's going to be the color of the flowers. So we're going to put 3 red flowers to 1 white flower. So if these flowers have 4 bikini, 3 of them are go red and 1 of them are go white. The rest of them are go. You get the point. And then the genotypic ratio is going to be 1 big R, 2 big R, little r, and then 1 little r. So that's basically the F2 generation, right? So as you can see, the first generation is usually very simple, really complex, everything 100%. So you're going to end up with 100% heterozygous. You're also going to end up with 100% of the dominant trait, which in this case was blue flowers. Now the dihybrid cross, which is a little bit more complex, right? So in a dihybrid cross, where you're going to be crossing two individuals, right? Which code for two different traits. So instead of drawing up that 2 by 2 squared little Punnett square, you're going to end up with a 4 by 4, right? So a 4 by 4. So we're going to end up with 16 boxes, right? 16 boxes in order to do a dihybrid cross. And the phenotypic ratio is going to end up with a 9 to 3 to 3 to 1. Genotypic ratio, you're going to end up with a 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1, right? So this is what is expected at the end of the day for the dihybrid. So then you have complete dominance, codominance, incomplete dominance, right? So you have that at the end of the day. So let's find out the difference between the three. So basically with complete dominance, It just means that only the dominant allele is going to be expressed. So for example, if you had two flowers, right? And we use a flower example, big R coded for red flowers, little r coded for white flowers. So the complete dominance means the offspring is going to come out completely red because that is the dominant trait. Incomplete dominance, neither allele dominates the other. So it means that if we had a white flower and we had a pink flower, no, let's just say we had a red flower and a white flower. If we had a red flower and a white flower and the fertilization they mated, let's just say that, and the flower that we're going to get is going to be pink flower. So it means that both alleles are going to be expressed, and usually when that happens, it's going to give rise to a completely new trait or a different color, because obviously neither parents were pink. One parent did red, one did white. So how did we get pink? Because it was incomplete. It was like a blend of both colors. For codominance now, both alleles get expressed. So for example, if a red parent or a white parent, they have offsprings, and that one plant, we realize that it have both yellow, it have both white, and it have both red flowers, right? That's codominance because both of them are expressed in that case. Anyway, so that's basically it. There is a biology series which basically has the nervous system, um, more detailed cell division, reproduction. Um, I can't remember the rest right now. Cells, cell structure and function, cell transport, different things like that. And then I also have the CXC playlist, right? Okay, so basically we're going to be doing a reproductive system crash course. So basically this is for both CSEC biology and the CSEC HSB. So even though CSEC biology is more general, and CSEC HSB is more focused on the human body, it's the same thing. It's the same thing in CSEC biology and it's the same thing in CSEC HSB. So for this particular topic, it's in both syllabuses, right? So you're gonna find the reproductive system in the CSEC biology syllabus, you're gonna find it in the CSEC HSB syllabus also. So you don't have to worry, say, okay, somebody something them you not have to know because you do CSEC biology, or somebody something them you not have to know because you do CSEC HSB. It's literally the same thing. The only thing is that CSEC biology students do a lot more in terms of plants and animals and all that stuff because it's more general. But CSEC HSB is focused on the human body, and in this topic we're talking about the human body. So basically the reproductive system is what we're going through. So we're going to try our best to go through this, explain it, and just to get an understanding and like an overview of what this topic is about. So if you never learn nothing, what I would advise you to do is take some notes. That is the first thing. Take some notes as me I go through the slides them. Read it as you go along as well. Also, so take some notes, read it, and stuff like that. So yep, so it's very simple. It should not be too long, I think, but anyways, we'll just get started. But yeah, so basically the goal is we want to get through these particular topics. So basically the reproductive system is very broad, right? There's a lot that goes into it. So what we want to talk about is sexual and asexual reproduction, the structure and function of our reproductive organs, both for the male and the female. Although, the female one I'm going to take up a bit more time because a lot more goes on there. Then you have the menstrual cycle, the birth process, birth control, and basically the importance of family planning, and then the issues relating to abortion. So yeah, this is just like a quick crash course, so we're going to take our time and we're going over everything right in a timely manner. So okay, so what I would say before we will get any further is to go ahead and like, comment, share, and subscribe. And then also you can check out this email right here. Check out the email right here if you want to turn for CSEC biology or CSEC HSB. And yeah, so let us just get started on this reproductive system topic. let's get into it. Okay, so if we're talking about reproduction, the first thing is that you have to know what reproduction even means. What does it even mean to reproduce, right? And especially in today's culture, sometimes we use the word so loosely, like, oh, reproduce, reproduction, oh, people just have baby and way. And if when we ask what is reproduction a while ago, if me said that and you say, oh, people have picnic, if that was the first thing that come to your mind, technically you are correct, right? You are correct. So basically, when we talk about reproduction, it's a process by which new offsprings are produced. And the word reproduction, it does not only go for human beings, it goes for animals. So animals, once you are living, you can reproduce. It is one of the 7 characteristics of all living organisms, the ability to reproduce. So humans reproduce, have offsprings, birds, dogs, fish in the water. Once it's living, it can reproduce. So just keep that in mind. And even though we're kind of focusing more on the human aspect, just know so reproduction go for everything. So even the plants, even our flowers, right? So we have to take into consideration that it's not just humans that reproduce, even plants need to reproduce also. So basically, there are 2 main types of reproduction. We have our sexual reproduction and our asexual reproduction. So off the bat, when I say sexual versus asexual reproduction, human beings don't undergo asexual reproduction. Let's just put that out there, right? We don't undergo asexual reproduction. Like, how would I say? So one example of a organism that can reproduce asexually are plants. Literally, plants don't need 2 separate genders, or yeah, we think, yeah, they don't need any gametes to basically fuse, right? In order to have offsprings, in order to have new plants being produced. Humans reproduce by sexual reproduction, meaning that both a male and female gametes have to fuse and fertilization has to happen in order for you to say that something undergo some kind of sexual reproduction. It has to fuse, male and female has to fuse, right? For asexual reproduction, it means that no fusion not take place, no fertilization took place. That is when you can say something is very much asexual. So human beings cannot undergo asexual reproduction, but plants can. And even though plants undergo asexual reproduction, they can also undergo sexual reproduction as well. So if you look at sexual reproduction right here, this is what humans undergo. That is how them have people have a baby. Male and female come together. You have the sperm cell, you have the egg cell, the sperm cells swim, go to the egg, fertilization takes place, boom. Somebody pregnant, and then boom, them have the baby. That's the offspring. That's literally the process. So yeah, we're going to go into a bit more detail about the advantages and the disadvantages for both of these types of reproduction. Okay, so basically, let us basically compare them more in depth. So basically, you have sexual reproduction and asexual reproduction. So basically, what we want to see are basically the disadvantages and the so basically both the advantages and the disadvantages of each type of reproduction. So in our sexual reproduction, obviously we said before that it involves 2 parents, fusion of gametes, fertilization, all that stuff is going to happen. Asexual reproduction, and you can literally note this down as a big difference. So if a question should ask what is the difference or name 2, 3 differences between sexual and asexual reproduction, this is what you can say. You can say that sexual reproduction involves male and female gametes. Asexual reproduction involves no gametes, sexual reproduction, fusion of gametes, asexual reproduction, no fusion of gametes happen whatsoever. And one of the biggest things when you're comparing asexual and sexual reproduction is that in asexual reproduction, what makes it so unique is that it have one parent involved, right? And what's so unique about asexual reproduction is that the offsprings are literally identical to the parent. When I say identical, it means them identical. You know, like twin, right? They're literally identical, like identical twin. The parents and the child that look like exactly the same. So with asexual reproduction, there is no variation. There is no variation at all. So that disadvantage for asexual reproduction is actually an advantage to sexual reproduction. So in sexual reproduction, they produce offspring that have variation. And you might say, why does it even matter if there is variation or not? You probably wonder. But variation is very important for a species, basically very important for a species species to survive basically because variation helps. It's like, how would I describe it? It's basically equipping the next generation to be able to withstand different conditions, right? So basically, it helps the species to adapt to new environments, help to reduce the spread of disease, can be used to increase food production. And the disadvantage to sexual reproduction is that it takes a longer time to produce offspring. So, for example, compare human beings. Compare human beings to what example could I really, So, for example, Hmm, I can't think of an example right now. What compare human beings having, call it say, all right, a pregnant woman have the baby in them belly, in her belly for 9 months. Is it that 9 months to give one or two child, right? Because the chances of having triplets and quadruplets, all of that stuff is very, very, very low. So say she have one baby or two in her belly at a time for 9 months, almost 1 year compared to plants. If the plant pollinate and whatever, the plant can grow up one entire area in that exact same time if you compare it like that. But look at it like this. So if there is no variation in asexual reproduction, right? And let's just say that there's a disease, right? And this plant is not, what should I say? It does not have the mechanism to fight off the disease, and the plant what a word now, susceptible, right? The plant is susceptible to the disease or whatever. And then that plant do asexual reproduction and produce 20 other plants that cannot fight off the disease. At the end of the day, all of them are go dead. All of them are go dead because basically the parent no good and all of the pitney them no good either. 'Cause all of them have the disease, right? So that is why variation is so important. So that is why variation is so important, and that is why human beings, you different from your mother different from your mother, and everybody. So even though you share the same genes, the genes not identical to the T like that. It don't identical to the T. It's not share some, but there is variation because you have some of your mother, some of your father mixed together. So if something already in your mother bloodline, we could have kill everybody. The thing is that fully enough your genes alone you carry. So maybe something in your father genes more dominant, and that is why it don't affect you. For example, sickle cell or something like that, right? It's not dominant, right? So it don't affect you, but you have the recessive trait for it or something like that. But you basically get the point when you compare them. That at the end of the day, they both have their advantages and their disadvantages. Variation is very important in sexual reproduction and in asexual reproduction. They can actually produce a larger number of offsprings in a shorter period of time. For example, we get the example about the plants. The plant produce 20 plants. In the time when one pregnant a human, for example, a human being take pregnant 9 months with one child or with two child maximum, typically. The time what it basically take to have that one or two picnic, the plants produce so much other plants during that 9 month period, like a lot. So you can basically see the difference. And Yeah, so the disadvantage of asexual reproduction is that they have an increased risk of disease and also no variation, and overall just they're kind of, what should I say, like a lower quality. It's almost like copy and paste, copy and paste, copy and paste. But what if there is something wrong with the parent? It means every offspring will have that issue as well. You get what I mean? With sexual reproduction, you never really know. Is either them have the disease or them don't. You honestly won't know because variation makes the difference in sexual reproduction. So basically, we're moving on to the female reproductive system. So I would suggest that you at least know how to draw this, right? I would suggest you at least know how to draw it. So basically, let's just go through the label. So basically, this is our reproductive system, and in here, right here, is the uterus. So the space in here is the uterus. So I feel like we've not started from the wrong place, but okay. So basically, this is where the vagina is down here, right? So this is the vaginal opening down here, right? So the vaginal opening, and then you have the cervix to the back. So basically, if for example you go to the gynecologist or something like that, this area is basically what they would be able to see from the opening back to the cervix. That's all they can see. And then up more is the uterus. So basically, uterus is very important. The uterus is very important. Like for example, when they say the uterus lining, for example, with the menstrual cycle, the uterus lining would basically shed, or basically yeah, it would break down. Uterus wall break down, like what I'm always saying. But basically, this is the ovary, this is the fallopian tube, and this is firmly, you don't really have to necessarily know this one right here, but everything else, the vaginal opening, the cervix, the uterus, the fallopian tube, and the ovary, I would suggest to draw this, or at least have a mental representation in your mind of how it exactly looks. So anyways, let's go over the functions then. So the first one is the vaginal opening. So basically, it allows sperm to enter the reproductive tract to potentially fertilize an egg for, and basically, yeah, so for reproduction, also menstruation, so it provides an exit route for the menstrual blood from the uterus. It also functions in childbirth, serves as a birth canal during delivery, and also for sexual function. But basically, everything goes on with the opening, like everything have to pass the opening right there. So if you give birth, it have to pass right here. So if menstruation, it have to pass right here. So this is very, very important. So now the uterus, which it is described as a pear-shaped muscular organ that don't spell properly, but yeah, that functions to nourish and host a fertilized egg until the fetus or offspring is ready to be delivered. So basically, it would be implanted on here, basically. And the ovaries, so basically, the ovaries are the female reproductive glands that produce the eggs and hormones that regulate the menstrual cycle, fertility, and pregnancy. And you have the fallopian tube, which its primary function is to transport an egg from the ovary to the uterus, right? So basically, the entire fertilization process would start with an egg being released from the ovary. The egg would travel through the fallopian tube. The sperm would wait in the fallopian tube for the egg. If the sperm and egg meet, fertilization takes place. So the fertilized egg, which is called a zygote, would travel down the fallopian tube. The zygote becomes a corolla. You're not really sure about what that means. Then a blastocyst as it travels to the uterus, and then it would eventually implant in the uterus lining where it can grow into a fetus. So basically, that is why the uterus is so important. And basically, if that implantation does not take place, that is when menstruation happens, right? So the body is preparing to have a baby every single month. So when the body is preparing to have a baby every single month, it is expecting, right? It is expecting that the egg, the fertilized egg, is going to implant on the uterus, right? So if that implantation does not happen, if it does not take place, then what's going to happen? The walls are going to break down, and it's going to break down in the form of blood, basically. So that's how the entire menstruation process happens, right? So then let's talk about the cervix. So it's a vital part of the female reproductive system. It has many functions, including fertility, pregnancy, childbirth, menstruation, and also protection. So as it relates to fertility, so the cervix allows the sperm to pass into the uterus during ovulation, pregnancy. So the cervix is what protects the growing fetus during the pregnancy, childbirth. The cervix dilates to allow the baby to pass through as in them a push out the baby, right? Like for example, if I hear them say, "Oh, 2 centimeters dilated" or whatever you want to say, that is basically what they mean. And then you have menstruation where the cervix opens slightly to allow menstrual blood to flow out of the uterus. Protection, the cervix secretes mucus to prevent bacteria and other harmful microbes from entering the uterus. And in recognition, the cervix recognizes and prevents pathogenic entry. Right, so you can see that the cervix plays a very big role. Okay, so basically this is the male reproductive system now, and basically just look at it, just look at it. So this right here, this tube is called vas deferens. You have the pubic bone, which is not really too relevant. Then you have the penis, the urethra here, the scrotum, the testicle, epididymis, the prostate, rectum, and then the ejaculatory duct. So basically, this is the male reproductive system. So you can just look at both of them and literally compare them. In this situation, it would seem like the female reproductive system is a lot more simpler, and the male one is a bit more kind of all over the place. But as I mentioned before, that's also seminal vesicle right there, but the pubic bone not really relevant, but everything else is really serving a purpose. Right, so this is a quick function for each of them. So testes produce sperm, the epididymis stores sperm, the vas deferens is what transport or conducts the sperm to the urethra, and then you have six accessory glands, which that one not necessarily there on that diagram, but it produce seminal fluid that nourishes the sperm. You have the urethra, which conducts the sperm to the outside, the penis, organ of copulation, the scrotum, provides proper temperature for testes basically, right? So it regulates the temperature. Right? So basically, when we say that the when we say that the penis is an organ of copulation, basically it means that it it basically transfers sperm, right? That would be the best way for me to describe it. It transfers sperm. So for example, when it transfers the sperm, it is basically what deposits the sperm into the female reproductive tract. So basically, that's what it basically means. Now, if we go back on the diagram, right, I will just look at it more in depth. So basically, the vast difference, we always pronounce it away, but the vast difference. So basically, it's a muscular tube that carries sperm from the epididymis. So this is it, the right of here. So this is the epididymis. So what the vas deferens does is that it carries sperm from the epididymis to the seminal vesicles, right? So look at it. It's carrying the sperm from here to the seminal vesicles, right? And then you probably say, what are the seminal vesicles? So basically, they are glands that produce a fluid rich in fructose, which nourishes the sperm and makes up a large part of the semen. So everything is working together in this reproductive system, right? And I would very much advise that you know at least one function for each of these, the relevant one there. Like the vas deferens, the urethra, testicles, scrotum, epididymis, the seminal vesicles, all of those stuff ensures you know the function, right? Ensures you know the function, but yeah. So we're with seminal vesicle. Yeah, so it makes up semen essentially, right? Then you have the testes. So basically, it's located within the scrotum, as we can all see right here. This is the scrotum, and it's basically responsible for producing sperm and the male hormone testosterone. So the sperm is produced, and the vas deferens transports it to the seminal vesicle. Seminal vesicle makes the semen, right? So we already get that far. And then urethra here is what carries urine and semen out of the body, right? And then the epididymis, and let's talk about the prostate. So basically, it's a walnut-shaped gland that surrounds the urethra and adds fluid to the semen, right? So that's really what it does. So we know that the seminal vesicle is what's going to make up the semen. You're saying that the prostate adds fluid to the semen right here, and then the scrotum is just a sack of skin that holds the testes and helps to regulate its temperature, right? And then this is pretty much self-explanatory, right? It's basically the opening where urine, semen, everything pass through the penis. So basically those are the functions, and then now the birth process. So the birth process. Okay, so the birth process, also known as labor, has three stages: the dilation, expulsion, and placental. So during labor, the cervix is going to open and the baby is going to move through the birth canal. So the first thing is dilation. So this is the first and longest stage of labor. And remember how me that say how you ever watch like maybe YouTube or some movie or something and you hear the doctor them say, oh, them say 2 centimeters dilated or 3 centimeters dilated, whatever you want to guess. But it's basically this. So this is the first part of the birth process. So basically the cervix is going to slowly open, and then during this stage, the pregnant woman would normally feel contractions, right? So they might be regular or irregular, or they might not even notice them completely. And as long as the doctor said it's safe to stay home, they would usually be at home during this stage. So like the early, like maybe 1 centimeter or 2 centimeter dilated, they don't need to be in the hospital, right? So when it start gets serious and they keep on, I guess having more contractions, more irregular contractions, or painful contractions, that is when them say, okay, we need to go to the hospital. So then expulsion, which is the second stage of labor, also known as the pushing stage. So basically, when you go to the hospital, well, not you, but when the pregnant person go to the hospital and then them check them and say, wow, them this much dilated. And then I say, well, listen, you already have the baby. So when them do that and them hook you up on the hospital bed and everything, or yeah, them hook you up on the hospital bed, put on drip and whatever you need and whatever. And depending on the situation, when them check the baby to see if the baby sit properly in the belly, yes or no, they will decide if you need a C-section or not. They will decide if you need an epidural or not. If them feel like they cannot push out the baby and all is going to be well, then I'm going to say just push out the baby. So yeah, so basically you go to the nurse or the midwife, I go monitor you and the baby, and then after them done set you up and everything, them I go tell you when it's safe to start pushing. So now once the baby's head is out, the rest of their body will eventually slip out during the next contraction. So basically, that is the ideal way for giving birth. Usually, them say make the head come out first because it's usually a lot more safer, right? It's a safer thing to do. If the baby sit upside down, sideways, across here, it will create a lot of problems. And in that case, they might tell the pregnant woman to have a C-section or something, or they might tell them to, I don't know, do something to try and turn the baby, right? So the placenta, which is the third stage of labor when the placenta is delivered, so this stage can take a few minutes or up to 60 minutes. So factors that influence, so basically that is when them push out the baby and them go, we call it cut the umbilical cord. But me pronounce it properly, them go cut the cord and all that stuff, and boom, we have a brand new baby. So that's the last stage. So now anyways, the factors that influence the length of labor, so whether you've given birth, whether the person has given birth before, or the frequency or the effectiveness of the contractions. So for example, if the contractions are just bad off the get go, bam 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 bam, then you go push out the baby quick and fast. If the contractions very if it's painful and the person is not like dilated very far, you're gonna sit down and pain and wait really and truly, right? So, and especially if the woman has given birth before, that gonna be a lot easier to push out the baby because at least our body did it before, so our body can do it again. If it's the first time, the body might, uh, whatever, but yeah. So, anyways, signs of labor with the backache, diarrhea, and the most obvious one, um, the water breaking essentially, right? We don't really know about them other one here and there, so let's just scrap that at least for now. Okay, so this is just the birth process again. So basically, the baby pushes against the cervix, causing it to stretch. Stretching of the cervix causes nerve impulses to be sent to the brain. Brain stimulates posterior pituitary to release oxytocin, and then oxytocin causes smooth muscle lining, but basically for the uterus to contract. So everything is a process. So when the contractions start, bam, 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 that is when you know the baby will come out. Then the menstrual cycle. So the menstrual cycle, it happens every month, prepares the body for pregnancy. If there's no implantation, there will be no pregnancy. So no implantation equals having a menstrual, yeah, basically. So basically it would happen. So technically they say it should be every 28 days, but it can vary from 21 to 35 days. So once the female falls within 21 to 35 days, it should not be alarming that much, but again, talk to your doctor. No, anyway, cycles often longer and less irregular for the first. No, they're often longer and less regular for the first few years after a person starts menstruating. Cycles tend to shorten and become more regular with age, and it can be light or heavy, painful or pain-free, long or short, and still be considered typical because pain being associated with it is seen as normal and somewhat basically it is very much normalized in today's society, right? About period cramps. It's normalized as if it's nothing, at least in today's society. So what happens during the menstrual cycle? So estrogen. So basically, once you're talking about the menstrual cycle, two very important hormones you have to talk about is estrogen and progesterone. Basically, so estrogen, when those levels increase, they cause the lining of the uterus to thicken, right? So the uterus lining is going to thicken, and then ovulation, when the egg is released from the ovary and moves into the fallopian tube. So now what the body is expecting at this point is, oh, I'm gonna get an egg will implant and you'll get pregnant. But when that doesn't happen, opposite happens. So next, the progesterone levels are going to increase after ovulation, while estrogen levels decrease. So then, if the egg isn't fertilized, progesterone and estrogen levels decrease, and the line of the uterus is shed. So that's what comes off as blood at the end of the day. If the egg itself is not fertilized, this is basically just a diagram of the process of the menstrual cycle, right? So these mountain or hill would basically represent it. It would represent the uterus line building up, right? And then obviously menstruation happens here. That is when it sheds and it goes down, then it starts build up all over again. And it happens again, type of build up, shed, build up, shed, build up, shed, build up, shed, all over again basically. Right? So there it is, line of the uterus, and it would shed the blood. Alright, then over here, the next topic is basically about birth control. So basically, this diagram shows the less effective to the most effective, and the most effective method would be abstinence, right? That's the most effective method. If one should choose not to do that, what's the next step? So basically, the next step is, and basically, these are the least effective: spermicide, fertility awareness, meaning that you would track the cycle to basically be aware of when you're ovulating, because if somebody is ovulating, that would be the best time. That's basically the time when they're most likely to get pregnant, right? For some reason. We don't know the science behind that, but whatever. Okay. Yep, so less effective. So this is 71 to 76% effective. And then this next round is 78 to 82% effective, depending on the method. So you have the cervical cap, the sponge, then condom. That one is external, and then you have the diaphragm. But me think this, and this would actually go inside of the vagina. And then 92 to 97% effective. You have the patch, the ring, the pill. I also have the shot. Then 99% effective. You have sterilization. Basically, this would mean that they tie their tubes, or them have an implant, or you get an IUD. But an implant is something them put in them arm, I think. I don't necessarily remember. But yeah, right? And then the next thing is the benefits of family planning. So basically, the benefits of family planning are the person would get to decide the number of children, and also the spacing of the children, right? So for example, some people want their kids like with a two year age gap, right? So they might say, oh, if you have one in 2024, you have the next one in 2026. So you basically plan it exactly, right? So it prevents unintended pregnancies. So, oh, once you did family planning, you can't say, oh, we never did know, or oh, we never did plan for this, or we can afford to take care of more pitney, right? So that is the benefit of family planning. So it reduces the recourse to abortion. So basically, and basically these are both connected, because one, unintended pregnancy, and then they say, oh, me don't want no pitney, me will get an abortion. So if you do family planning, you won't get pregnant. Because if you know you don't want no pitney, it makes sense you'll get pregnant, if that makes sense. Right? So reduce giving you the opportunity to go get an abortion. And then getting an abortion can have risks to it. So then also family planning decrease HIV and STIs, gender equality and education for women. So that basically the person is not necessarily, I'm just saying, no, just to have pitney back to back to back when they can't take care, and they actually get to go to school, and then get like them other life goals out the way as well. And this one is for females specifically. And then slower growth reduces pressure on resources. So basically, once you do family planning, you're able to plan a lot more. For example, if you know you can't take care of 5 pitney, it means you're not gonna have 5 pitney, right? That's basically what it means. And then cost savings as healthcare is by, right?
So basically, this is just healthcare because an idea picnic expensive. Children are expensive, so it goes back to resources. If you cannot afford it, don't go down that road. But when you have five picnic, the five picnic, them sick on you, how would that go? Right? And you plus your health insurance and stuff like that. And then it saves the lives of women and newborns. For example, you're having let's just say you have a situation, maybe rape or something, and maybe a 12-year-old get pregnant, and because the 12-year-old body is not, even though technically yes they can get pregnant, their body don't obviously no, it's not fully developed to bring a child. So now when we get pregnant with a child and that 12-year-old could die with the baby. The baby didn't she did too because her body not strong enough to push out no picnic. Right? So basically those are the benefits of family planning. Right? And then the next thing that we're going to look at is the issues relating to abortion. So there are ethical concerns, right? So some people might say it's wrong. It's just wrong to kill the fetus, kill the child, and all that stuff. So there are ethical concerns, legal aspects, right? And you know that abortion is not legalized everywhere, right? In some countries, getting an abortion is very much illegal. And if them find out you get an abortion, you can go to jail, basically. And then the medical risk, right? Because when people do abortions, especially when them do it under the quiet, like some local warehouse back up with something, it put them at risk for infection, excessive bleeding, incomplete abortion, meaning the local person what they want to do because for example in countries where abortion is illegal, let's just say them go somewhere under the counter, go try and get the abortion done. It has risk to it. And let's just say the person not do it properly or them when they want to do the abortion, them left something inside of them, like when it's supposed inside of them, infection and basically kill them off, right? Or mess up the urinary tract or whatever you want to call it, right? Right. So then there's also the social stigma because people usually look down on women or people who decide to have an abortion. That's one thing. Access to care. So depending on where they're from, especially somewhere like when you want to say Africa, maybe somewhere in a maybe Israel. I know they're local country, they're not in places like Israel or them places. In places like that, it might be hard to, you know, seek help or whatever. We don't know if it's and then impact on future pregnancies, that's also a risk. So basically what we went through sexual and asexual reproduction, destruction, function of the reproductive organs, menstrual cycle, the birth process, the birth control, the importance of family planning, and the issues relating to abortion, basically, right? So basically this would be the end of the reproductive system process or the topic. However, right, so it'd be the end of the topic. But yeah, take everything with a grain of salt in it, right? But basically, these are the main areas that would fall under the reproductive system. So what you need to do is just know about these things, know about the differences, the advantages and their disadvantages, the structure and the functions, be able to explain the menstrual cycle, or if you're given a diagram, be able to interpret it, the birth process, the birth control, different methods of birth control. You have the importance of family planning and then the issues relating to abortion, right? So these are all of the topics that we went through today.
Okay, so basically now we're going to do the circulatory system. So basically this goes for both the Csec HSB as well as the Csec biology. So if you're doing either Csec HSB or you're doing either Csec biology, it does apply, right? It's the same thing. It is literally the same thing. So basically, that is what we're going to get into. And basically, when we talk about the circulatory system from the get-go, basically what it's saying is we're trying to we know that organs make up systems, right? So the order is that cells make up tissues, tissues make up organs, and then organs make up systems. So that's why we're talking about the circulatory system. So we're talking about how blood gets around the body, how it gets oxygenated, and basically how that entire gas exchange process takes place to keep to basically keep the blood having oxygen in it. So that's what we mean by circulatory system. So yes, and we're going to be talking a lot about the heart and all those things. So yeah, so the topics that we're going to go into are basically why do we need a circulatory system. That's one. The materials that are transported around the body. Then we're going to be talking about the components of the blood. Then we're going to be looking at the structure of the heart, like the design, or the the outline, the structure of the heart basically. Then we're going to be talking about blood vessels such as arteries, veins, capillaries, and basically the role that they play in all of this. And then we're going to actually discuss exactly how double circulation takes place, and basically why it takes place in our body. So basically in humans we have double circulation. So basically what that means is that the blood passes through the heart twice. So obviously deoxygenated blood, and then we're going to have oxygenated blood, right? And then we're going to look at blood pressure, meaning systole and diastole. So that's basically the plan with the things that we are going to get into. Okay, so the first question that we're going to answer is why do we need a circulatory system, right? So why you think we need a circulatory system? Why we never just alive and just there? Like if we compare something like maybe a flatworm or something really small, those organisms that are really small, they don't really have circulatory systems because they don't need it, right? They don't need it. They're small enough. The surface area to volume ratio, it's good enough. They have enough surface area to diffuse and do everything that they need to, right, in those small organisms. But in human beings we are big, right? We are big, like literally we have so many systems going on in the body. We have obviously the circulatory system, excretory system, respiratory system, reproductive system. We have a lot of systems, we have a lot of things going on. And basically when we're talking about the circulatory system we have to talk about the heart, right? So the heart is literally what pumps blood all over the body, right? The main function of the heart pumps blood all over the body, right? So every other organ needs blood. So that heart, right? If we lose our heart, basically you're dead. You're dead. If you don't have a heart, you die, right? For example, and if we look at another system, like for example, maybe the reproductive system, let's just say a woman takes out her uterus, she can still live afterwards, right? But with the circulatory system and with the heart, there is no living. The two biggest organs, yes, if you lose your brain and if you lose your heart, you're done for. There's no going back after those two have been damaged. And obviously some people can get the thing put in them chest. I don't remember what we call right now with the heart, the metal heart. I don't remember what it be, but basically I don't remember right now, but yeah. So it's very essential now. It literally now the circulatory system, it literally delivers essential oxygen and nutrients to cells throughout the body while removing waste products like carbon dioxide. Now, we know that oxygen, we as human beings, we inhale oxygen and we exhale carbon dioxide, right? So every single time we inhale that oxygen, every single organ in our body needs that oxygen, right? We need the oxygen in our blood because the blood is obviously what circulates, right? The blood is what circulates throughout our entire body, right? And we need that, we need to keep that oxygenated, right? So every single time we exhale, we're removing the waste and we're getting the carbon dioxide out of the body, right? So with the circulatory system, as I said before, this is why we need it, right? One, we're just too big as organisms. We're big. Like the surface area to volume thing would work with at all. The surface area is way too big. There's no way to just diffuse out carbon dioxide out of our body and just, there's no way to do that, right? So that is why. the circulatory system is so important. That is why the heart is so important, because it is what drives this entire circulation process. Both the heart, the blood, and obviously carbon dioxide and oxygen. Those are the two main gases, right? So now some different materials that we transport around the body. Obviously, oxygen and carbon dioxide, the two main gases that we just talked about. Hormones. So basically, we're not talking about the endocrine system right now, but we know that with nervous system they use nerve impulses. With the endocrine system, they use hormones in order to get things around the body, and hormones work through the bloodstream, right? So you can just see how the circulatory system is just so important and how it kind of overlaps with other systems, because other systems also depend on the circulatory system in order to work, right? And if we're talking about circulation, as I said before, blood. The blood is the driving force behind the circulation. It is literally what circulates throughout the entire body, right? And when we talk about blood, we're obviously going to look at the components, and we're also going to look at blood vessels, because the vessels, those are like the containers, those are the tubes that the blood is going to travel through in order to distribute slash circulate throughout the entire body, right? So let's talk about the components of blood, and it's very important that if you do the circulatory system, you need to know the components of blood, right? So basically, what we have going on is the plasmid, right? So the plasmid is basically like the liquid part of the blood. It makes up about 55% of its volume. It is mainly water and contains various dissolved substances like proteins, salts, glucose, hormones, and waste products, right? But the main thing that you should never forget about the plasma is that it is the liquid part of the blood, right? The liquid part is like make up 55% of its volume, right? So if you ever like bleed, maybe you cut your finger or something and you see the blood, so the liquid part, the liquid aspect is its plasma, right? Now, red blood cells, and obviously they contain hemoglobin, right? And we know that when we hear hemoglobin, we know that hemoglobin and red. I don't know, but me personally correlate. Whenever I hear hemoglobin, I correlate it to red, right? So the reason why the red blood cells and the red blood cells are the next component of the blood. So these red blood cells, right, that contain hemoglobin, and that hemoglobin gives the red blood cells its color, right? So that is why blood is red, because hemoglobin inside. And the next very important thing about red blood cells, right, is that they carry oxygen. So literally, if you ever hear like them say some people anemic or them have low blood count and stuff like that, but basically it's very important that we maintain a healthy level of red blood cells, right? Because they are literally the oxygen carrying cells within our bodies, right? So they carry oxygen from the lungs to different tissues all over the body. So they are extremely important, right? So hemoglobin, so what you need to take away from red blood cells is that they carry oxygen, they're made up of hemoglobin. Hemoglobin gives it its red color, right? So basically plasma is in the blood, red blood cell, they're all these four, they are all inside of the blood. So when you bleed and you just see it like that, your naked eye can't really see much, right? But put it under a microscope and these four main components of blood will be literally seen, so you'll be able to see everything. Now this word right here, eh, I can't even pronounce it myself, but erythrocytes, this basically is the same thing as red blood cells. It's just another word for red blood cells, or the more technical name for red blood cells. But just remember that plasma, liquid part of the blood, red blood cells carries oxygen, right? It carries oxygen and it contains hemoglobin. So just remember those main things about these two. So now white blood cells. So the main thing that you need to remember about white blood cells is that they fight infection and they fight diseases, right? So for example, if you get a cut, right? If you get a cut, these white blood cells are literally what would rush to the area, right? So basically, if as we said before, we get a cut, white blood cells literally what fight infection and diseases. So they try their best to prevent you from getting sick. They are part of the immune system, right? And then being a part of the immune system, that is their role. Infection, fighting infection, right? So as you can see, within every single cell, so the four cells that we're talking about, right? And this is the third one, right? Blood cell. You can realize that each of them, each component of the blood, carries out a separate role. Like they're not overlapping in function, and you understand exactly why we need the plasma, why we need a red blood cell. I know you're seeing why we need white blood cells, right? And just to make it clear, white blood cells don't actually white. And they don't actually like just white. If you look at it under the microscope, it's not going to be white. You understand? But I don't know why they call it that, but it's white blood cells, right? So the last one we're going to look at is platelets. So basically, the main thing about platelets is that they function for blood clotting. So they clot blood, right? So helping to stop bleeding when a blood vessel is damaged. So as I said before, if you get a cut or anything like that, this is what helps the blood to clot. You understand? It helps the blood to clot up together to stop bleeding. So as you look at them, you just need to know the function and know that these are the components of the blood. You might see it on the MCQ, or you might see it on the paper too in a free response. So infection, white blood cells, and then blood clotting, platelets, right? And the thing about it, so the leukocytes thing, you don't have to really worry about that, but leukocytes is another name for white blood cells, and thrombocytes and then is another name for platelets, right? So it's the same thing. Then now, in order for the blood to circulate all over the body, we have different blood vessels, right? So we have different blood vessels, and they are arteries, veins, and capillaries, right? So in their structures, they are very, very different. Very, very different, right? And there's a photo right here comparing the three of them. So we can see an artery, we can see the vein, and then the capillary. So usually the artery is the biggest one, right? It's the biggest one, right? So the arteries, they carry oxygenated blood from the heart to the rest of the body, right? So just remember this: arteries carry oxygenated blood from the heart to the tissues and organs of the body. So it means that they carry oxygenated blood away from the heart. So away from the heart. No, it's saying here that they have thick elastic walls to withstand high pressure of blood pumped from the arteries, right? So the high pressure of blood pumped from the arteries, right? And they also branch into smaller vessels called arterioles, which then lead to the capillaries. Then they have like venules and then the vein, but you don't have to necessarily worry about all of that. But that's just how it travels. You have the artery, which is the biggest, and you have arterioles, which are going to be smaller vessels, and they got the capillaries in the middle. Now I have the venules and then the vein. So as I said before, the arteries are usually the biggest blood vessel. Let's just say that, right? No, one of the most noticeable things about the artery is the size of the lumen. So this is actually very important. So the size of the lumen, you can see that the lumen is quite small. So we're saying that the arteries carry oxygenated blood away from the heart to the rest of the body, and it has a small lumen. It has a small lumen because, and basically lumen is like a little hollow space, the space, right? So, and that's the space where the blood will travel through. So now it has a small lumen because of the high pressure of the blood that's being pumped from the heart, right? And you know, so the more narrow something is, the faster the pressure will go, right? So it's like if you have a pipe, right? And if you have a small pipe or maybe a straw, we can probably use an example with a straw. Let's just use the example. For example, you take a straw, and we're going to say that we're going to throw some water in the straw, right? And we're going to say that basically we go through the water in the straw, and the thinner the straw, or the mager, or the skinnier, the narrower the straw is, the water will come out quickly. Now if we use a big broad straw, it just kind of it basically has lower pressure. That is what me I try to say. The bigger it is, the lower the pressure, and then the more narrow or smaller the lumen is the faster the pressure. So the blood is moving fast. So it's narrow and the blood does not move fast. So it'll leave the heart and it'll go to the the rest of the tissues and organs of the bodies. Because remember we said that all the organs them, all the other systems them, all our tissues that depend upon this oxygenated blood, because the blood is what circulates all throughout the body, right? So the blood circulates to the reproductive system, all the endocrine system relies on the blood as well. So as you can see, it all over the place, right? So that's basically why the lumen is so small in the arteries, because it is carrying higher pressure of blood that is being pumped from the heart. Now we're going to take a look at the vein, because that's important too. So in the veins now, they're bringing deoxygenated blood. So remember that arteries carry oxygenated blood away from the heart, and in veins are going to bring deoxygenated blood to the heart, back to the heart, right? So now the same thing that we were saying is that, as you can see before, as you can see here, that the walls are much thinner in the veins and the arteries, right? And they have valves. So now valves in our veins are basically, they're very common, and they serve a very key essential function, right? To prevent the backflow of blood, right? So we say veins are carrying a deoxygenated blood to the heart, right? And then though these veins have valves to prevent the backflow of blood, because we don't want the deoxygenated blood out, as to go where it's not supposed to. So now, as you can see now, the vein has an extremely large lumen. Why you think the vein have thinner walls, and why you think the vein has such a big lumen? The same thing that I said before, that when when the more narrow something is, the faster the pressure of the blood. Now we have a big vein, we have a big lumen, have a big space in the middle, so it means that the pressure is much lower, right? The pressure is much lower. So basically, the blood is now moving back to the heart. So imagine that. Remember, I said the heart pump blood, So for the arteries, them are easy something. The arteries can just take with the blood quick and fast, right? That is why they have so narrow lumens. Now in the vein, this blood is going back to the heart, right? It's going back to the heart. So you cannot have the blood just rushing pan the heart just like that. Remember, I said the heart is an organ. Remember. So when the blood was leaving, it was fine. You can have the pressure of the blood go as fast as you want it to go, because it go to the organ, it go to the tissues in the body. But when it comes to the heart is just a small little structure. You cannot just have blood rushing upon the heart like that. And that is why we have such a big lumen. We have a larger lumen, and this lumen facilitates like the slower pressure of blood towards the heart. So a much lower pressure of blood towards the heart, just to avoid any complications or anything like that, because you cannot just have blood rushing in upon the heart if you get damage, like, you understand? So just keep that in mind. And in our capillary, which is, you can see there, it also has a small lumen, but basically so the key thing is that the capillary, they are only one cell thick. If you compare the artery, the capillary, and the vein, it is the only one that has one cell thick. Just look on it, only one cell thick. And the reason why it's only one cell thick is because it functions in the role of gas exchange, right? So this is the key gas exchange blood vessel, the capillary. So it is literally the middleman between the artery and the vein. This is literally, so the artery I bring blood away from the heart, and the vein I bring blood to the heart. But the capillary is literally that middle ground between the artery and the vein, where the gas exchange is going to actually happen, where they're going to remove carbon dioxide, which is a waste, and then take in oxygen and nutrients that we actually need. And then that is what's going to get circulated around the body and all that stuff, right? So it's one cell thick, and just know that its primary function is for gas exchange. So just remember that part. Yeah, so basically it's just surface area. So that's why it has such a small surface area, because it's one cell thick, and obviously the fusion works best through shorter distances, goes through thinner walls and stuff like that. So that is why it's like that. Alright, so this is just the diagram of the heart, showing the different parts of the heart, right? And we basically soon discuss what is happening with everything right here. But just to get an idea, just look at all the different parts. We have the superior vena cava, we have the right atrium. It would be good if I can draw this, right? Very good if I can draw this and just know the different parts on it. So you have the superior vena cava, you have the right atrium, the um, right ventricle, left atrium, left ventricle, pulmonary artery, pulmonary vein, the mitral valve, aorta valve, pulmonary valve, tricuspid valve, which prevents the backflow of blood. So all of these parts of the heart are very, very important. And we're going to talk about how all of this works in just a second. So, we said before that we have, as humans, we have a double circulatory system, right? And we said that blood passes through the heart twice. That is what we said. Now, why is it so advantageous to have a double circulatory system? So, more oxygen and nutrient delivery, improve the gassing exchange, and the ability to maintain higher metabolic rates. As you see, all that physical activity and run up and down business, it works out. And as I said before, because of the size of our bodies, we need this double circulatory system. The blood cannot pass through the heart once. It now goes to sustain the body. A lot goes on in the body. We have a lot of tissues, we have a lot of organs, we have a lot of systems going on. I'm gonna tell you about the reproductive system, nervous system, endocrine system. A lot of things are depending on the circulatory system. We need the blood, we need the oxygen for flow in our body, right? So this is why the double circulatory system is necessary, and it's called that because it passes through the heart twice. Right? So now just bear in mind that, and if you look at this diagram, just bear in mind that when the deoxygenated blood, right? The oxygenated blood, it gets oxygenated in the lungs, right? So from the lungs, that is where it gets the oxygen from. Just know that, right? So now, as relates to circulation, we're going to talk more about that. But basically, what's happening is we're going to focus on this diagram right here. And basically, the deoxygenated blood is going to return to the heart through the vena cava. So right here, this is the vena cava. So deoxygenated blood is coming in. It's going to the right atrium, then it's going to the right ventricle. So this tricuspid valve is just what prevents the backflow of blood. So it just prevents blood from going the wrong way. We don't want the blood going back out through the vena cava. We want it going in one direction, right? So it comes to the right ventricle, where it's pumped to the lungs via the pulmonary artery. So as you can see, this is the first lap of the blood, the circulation. I would say blood passes through the heart twice. So see there, this is where deoxygenated blood comes in, and then it's going to be pumped to the lungs via the pulmonary artery. Right, good. Then we're saying oxygenated blood is going to return through the pulmonary veins, right? So it's going to return to the pulmonary veins, go through the left atrium, and then go through the left ventricle, and then it's going to be going out through the aorta. So after the gas exchange takes place in the alveoli in the lungs, that oxygenated blood is going to come back. Right? So when it comes back, it's going to enter through the pulmonary veins and then go to the left atrium, right? When it goes to the left atrium right here, it's going to enter the left ventricle, and then it's going to pump oxygenated blood into the aorta, and then the aorta to the rest of the body, right? So all these different organs and tissues that need this oxygenated blood is going to get it, and that is how we say that blood goes through the heart twice, right? So this deoxygenated blood returns to the heart with the vena cava, completing the systematic circuit, right? So as you can see, it's a cycle. So the pulmonary circulation, it starts out deoxygenated blood coming to the vena cava, right atrium, right ventricle, and then out through the pulmonary artery to the lungs, right? So that's what was it happen. Obviously, in the lungs, you have tiny air sacs called alveoli. Gas exchange takes place, so then oxygenated blood comes back in through the pulmonary veins. So the pulmonary veins, they're bringing oxygenated blood, right, to the heart. In this case, remember this is different from the regular vein. The regular vein, them, they bring deoxygenated blood to the heart. That's what the regular veins do. So just look at it like this. Regular veins would bring deoxygenated blood. Alright, so now with blood pressure, basically we know that blood pressure is hypertension, right? So basically we're going to be talking about basically how they measure blood pressure. So basically you have the systole value and you have the diastole value. So basically the systole is the phase of the cardiac cycle when the heart contracts and pumps blood out of the heart, right? So that is that aspect. Then the diastole is the phase of the cardiac cycle when the heart relaxes and refills with blood. So if you look at the diagram, the top number, the bigger number, is going to be the systolic number, right? Which basically tells when the heart contracts and pumps blood out of the heart. And then the diastole is when the muscles are basically more relaxed, right? So basically, pressure exerted when blood is injected into arteries when they're contracting. So basically, it's saying the normal value for an adult would be 120, and then for the diastole value, 80 would be the normal value, right? So that's basically what it means. Systole and the diastole, the big top number, systole, the bottom number is diastole, which is usually a smaller number, and 120 over 80 would be a regular blood pressure for a relatively healthy human being. So if you see the systole number is like 140, 150, then you say the person have high blood pressure. And yeah, so basically that's the circulatory system. Ensure that you know the parts of the heart and basically how blood goes in and how blood goes out. So basically we're going to look at the excretory system. So basically we did the reproductive system, there's also the nervous system, the circulatory system, there's different systems, just overall a part of the crash course. So the different types of cells, cell transport, and all of those things. A little bit about photosynthesis and obviously hereditary and variation, all them something there. We did look on that. So basically the whole mitosis, meiosis, and how genes get passed on and stuff like that. But now we're going to look at the excretory system. So basically, the excretory system is probably going to be one of the shortest systems. But basically, we just go through the main points. So it's for both CSEC biology as well as CSEC HSB, right? So it's basically the same thing, right? It's the same thing I need to know for both subjects, but basically, in my memory, remember some years ago, right? We did actually see, or actually, me feel like it was the year that me did do CSEC HSB, the year me do that subject then. That was the year me think them put something relating to the excretory system and wish me did surgery, but that would be what the CSEC HSB, the 2021 exam, the paper 2 for 2021. So as me look on this now, me I say wait, me did actually remember when me do the exam, there was a question relating to the excretory system, in the sense that they were basically, I think it was a scenario about one man where drink and stuff like that. I don't remember exactly what the question was, but as you can see, especially for HSB, this excretory system is going to come in handy, especially when they give you like a scenario and you have to know how the excretory system works. You can say, oh, this is what's happening based on what the scenario tell me, say, right? So basically, it's important, especially for HSB, when we do biology, we never really see the excretory system come on the exam like that. Obviously, it is a part of the syllabus, right, for CSEC biology, but we never really see it pop out like that when we do CSEC biology, because obviously CSEC biology is a lot more broader, and then CSEC HSB is more zoomed in on the human body, right? So that is why it would show up more in HSB. But yeah. So anyways, this is the plan. We talk about what is excretion and its importance, the roles of the organs that secrete, that not secrete but excrete substances in the body. Don't mix up secrete and excrete. Like when something excretes, we're talking about waste. Just keep that in mind, like waste we're trying to get rid of. With the word secrete, kind of work more with when you talk about hormones or stuff like that, things that actually serve a function in the body. That's why something secretes something, right? So like for example, in our stomach we secrete enzymes, digestive enzymes that act on the food we eat and stuff like that. But don't mix up excrete and secrete, because they're two different things. And when it's a secrete, when something secretes something, more than likely it serves a function. When we're talking about excretion and the excretory system, we are focused on strictly waste, right? Strictly waste, something that the body just does not need. We are trying to get it out. So, and then obviously the structure and function of the kidney. So we're just going to look on the kidney a little bit. Yeah. But I feel like I'm gonna mention something else. What is my mind? I feel like, what was I saying about, yeah, so we'll just continue and hopefully remember what I think about. But basically, excretion, so it is a process by which organisms eliminate metabolic waste products and other toxic substances from their bodies, right? So excretion, if you remember Grimner, right, as in growth, irritability, that meaning sensitivity, movement, nutrition slash feeding, and then excretion and but it's a reproduction or respiration already, but you get the point. But yeah, if you remember Grimner, you know that excretion is one of the characteristics of living organisms, right? And the fact that it's a characteristic of living organisms, this can be an indication that something is living. So in order for something to quote unquote be living, it has to not secrete but excrete waste, right? Because within our bodies different metabolic processes are taking place, right? And with these different metabolic processes taking place, we're going to have waste products, right? Even our sweat, different things. So yeah, for example, is our sweat and obviously urine and then feces, things like that. Even menstrual blood, that's also waste, right? So there are different methods or ways that we excrete things from our bodies, right? So yeah. Then basically the main purpose behind excretion, as I said before, we're trying to get rid of waste outside of the body, right? We're trying to get rid of waste. But the thing about it, if we're trying to get rid of waste, why is getting rid of this waste so important, right? And this is because of two things, well, two main things. One, we're trying to maintain homeostasis in the body, that's one. And then two, we're trying to prevent the buildup of harmful substances, right? So homeostasis is a very important thing. Is basically maintaining the internal environment. So basically, regardless of what is happening around you or around your body, or basically regardless of the temperature or whatever is taking place, it's important to maintain homeostasis, right? So it's like a constant, balanced internal environment despite external changes, right? So for example, if you go somewhere that's cold, and obviously we'll start to feel cold, because if the environment cold, we're gonna start to feel cold. But when we start to feel cold, our body's response is to shiver. So when our bodies start to shiver, that's the response that, okay, we need to maintain homeostasis, because guess what, if we stay in the cold, we're going to freeze, right? We're going to freeze. Cells and different enzymes in our bodies cannot function in that at that temperature, right? And so that's why we wear sweaters and different layers and different things like that in order to maintain that internal environment at the end of the day, right? So homeostasis is a very important part of just human beings, right? And then the next point behind excretion is to prevent the buildup of harmful substances. So basically, why is this important? So to prevent the buildup of harmful substances, it's important because one, it can damage tissues, right? So we know that tissues are made up of cells, so that's that. So we don't want to damage tissues, we don't want to damage organs, we don't want to just disrupt different bodily functions, right? Because let's just say, for example, you have the waste in your body, and like for example, maybe somebody have cancer or something like that, and maybe your colon or something like that is affected, and the waste is not getting out of the body. What can happen is that the waste can start to we call it, it's more like it kind of, why we just feel like we define the word secrete. What is not secrete, I mean to say, it's more like it can seep into the bloodstream, right? And if that waste gets into the bloodstream, you're in problem, because blood, remember the blood, if you remember the circulatory system, blood is what travels all over the body, right? It is what circulates the entire body. It is what basically works with every system within the body, right? And we say that organs are what make up different systems, right? So imagine the waste getting into the blood, and then okay, endocrine system completely mess up now, because remember the endocrine system is supposed to work with the hormones. Now hormones, they travel basically within the bloodstream. So that mess up. Circulatory system mess up too. So basically the whole double circulatory system with the heart problem start there so too. um, Everything can just get really thrown off in the body if there's a buildup of substances, right? It can become toxic, and it can basically kill you if you don't get rid of waste out of your body. And as I said before, it's done through the process of excretion, right? So this specifically goes for human beings. Obviously, like different organisms, they would use different mechanisms, right? So maybe something like what is it called, like an amoeba or something like that, they would use like exocytosis to like get stuff out of their bodies, right? So but we're focusing primarily on the human body. Okay, so basically now as it relates to excretory organs, right? So the main organ that we're really focusing on is the kidney, right? So basically, the kidney is the main excretory organ, and it is what filters out blood and allows the waste to pass out through the urine, right? So even though the kidney is the main excretory organ, we also have other organs within our bodies that secrete different things. Not secrete, but excrete, right? So basically, we have the lungs, obviously, our entire respiration process, how we take in oxygen, and then we exhale carbon dioxide. So the lungs do do serve as excretory role. Our skin, because when we sweat, and our sweat is made up of like water, salts, ureas, just different things, right? That's also helping to get rid of waste from inside the body. That's usually why when you sweat afterwards you're thirsty, because you lose water and you also lose salt, right? So that's another important excretory organ, the skin. And I always say that the skin is the biggest organ, made up of three layers: the epidermis, which is the outermost layer, the dermis, which is below the epidermis, and then we have the subcutaneous layer, which is the bottom layer, which is made up of primarily fat cells, right? So even though the focus is on the kidney and it is the main excretory organ, we also have the lungs that serves its role. We also have the skin that serves its role, right? And basically, just to know that the urethra removes urine, right? So basically, the kidney is what would filter out the blood and make the urine, and then the urethra is where the urine would actually pass out to get out of the body, right? So as it relates to the structure and the function of the kidneys, so as I said before, it is the main excretory organ, right? It is the main it is the main excretory organ, and if we're focusing on the structure, or basically you just need to know what it looks like, but basically it's two like bean shaped organs, right? So the kidneys, we don't have one, we have two kidneys, right? So a normal healthy human being would have two kidneys, and obviously you have people who um different illnesses, they only have one kidney, or maybe somebody donate a kidney to them, different situation. But a healthy normal typical human being would have two kidneys, right? Whether or not both of them functioning or not, they won't know unless they do a test, right? But basically it filters out waste from the blood, and we talk about why that's so important, because it can literally kill you. All that build up in your body can literally kill you, right? So it can help to regulate blood pressure, fluid balance, and also electrolyte levels, right? So if we're just going to look at how it's shaped, so we have two kidneys, a kidney on the right and a kidney on the left. So basically that's the structure of it, right? And now within the kidneys we do have some structures which are nephrons, right? So basically nephrons would be like tiny filtering units within the kidneys that remove waste and excess fluid from the blood, right? So that's the like overall that's the main thing with the kidney. You have to know about the nephrons which are inside of the kidney, since they're the ones that actually do the filtering, right, in order to remove the waste. So it's not just the kidney that remove waste all by itself, it's the nephrons that's within it that's actually removing the waste and the excess fluid from the blood, right? So now we said urethra is actually what carries the urine outside of the body, right? And then if we look at it now, and then the bladder would basically be the sac that stores urine until it's excreted, and then the ureter would be the tube that carries the urine from the kidneys to the bladder, right? So the urethra is carrying the urine from the kidneys to the bladder. You can obviously see the bladder, and then you can obviously see the bladder here, and this is the urine inside of the bladder. And basically the urethra would be like the final place before it gets the urine outside of the body. So kidney, we have our tiny filtering units, which are basically the nephrons. Actually see there the nephrons inside of the kidney. They filter out the urine. The ureter will bring it to the bladder, and then the bladder would store the urine. And then when you feel like, oh, you want to urinate, when you actually go to urinate, the urethra is where the urine would pass out through, right? So it's actually very simple. Like when you really look on it, very simple. Just know that is the kidney. We have in nephrons. This is what the nephron look like. The ureter, if you want, just follow along with the diagram. But use the ureter, right? And do not mix up ureter and urethra, because I feel like when we learn it, we used to mix them up, ureter and urethra. Just remember, say urethra, the way down at the bottom is literally the exit at it to bring the urine outside of the body. So you gotta work with the kidney, right? And inside we have the nephrons, and then the nephrons are the tiny filtered urine units, and then the ureter would bring the urine from the kidney. So after the nephrons unfiltered, bring the urine from the kidney, go to the bladder, right? And then the bladder would store the urine. When you decide that you actually want to urinate, it's going to pass out through the urethra, right? So that's basically the structure and the function of the kidney, right? So it's actually very simple. Just look at the diagram and basically follow the diagram. And in this example, as you can tell, this is basically a female body, right? But it's almost the same thing. Almost, except in the male, obviously you won't find a uterus and stuff like that. But yeah. So this will basically be the last thing, right? So how urine is formed. So we said that the kidney has basically nephrons, which are tiny filtering urine units, right? So these tiny filtering units, which are inside of the kidney, how do they form urine, right? So they're filtering the blood, right? That we know that we just need to know that these nephrons they filter out the blood and that waste passes out as urine. We just need to know that from the get-go. So this diagram is very simple and we're just gonna follow it in order, right? So first we have the glomer glomerulus, right? With this bad pronunciation, but glomerulus, right? And basically it filters out the small solutes from the blood, right? Because remember, this is the whole role of the excretory system to filter the blood. And the end product is going to be urine. The urine is going to be the waste that is getting outside, getting out of the body. So the glomerulus is what's going to filter out the small solutes from the blood, right? And then the proximal convoluted tubule is going to reabsorb ions, water, and nutrients, and is basically going to remove toxins and adjust the filtrate pH, right? So it's going to filter first, then it's going to reabsorb ions. So it's basically a filtering process. Filter out the small solutes, then it's going to reabsorb the ions, water, nutrients, remove the toxins, adjust the filtrate pH. Then in 3, you have the descending loop of Henle, right? So descending loop of Henle, and basically aquaporins allow water to pass from the filtrate into the interstitial fluid, right? So that's going to happen, and then in the ascending loop of Henle is going to reabsorb sodium and chloride from the filtrate into the interstitial fluid. And then the distal tubule is going to selectively secrete and absorb different ions to maintain blood pH and electrolyte balance, right? And then you're going to have the collecting duct, which is going to reabsorb solutes and water from the filtrate, right? So it starts with the glomerulus, then the proximal convoluted tubule, and then the descending loop, descending as in going down, loop of Henle, then the ascending loop of Henle, and then you're going to have the distal tubule, and then you're going to have the collecting duct. So basically, this is just something more of a like a memorization thing. So you just need to memorize what happens in what, right? At the end of the day, the goal is to filter the blood and to pass out the waste through urine, right? That's the whole goal of it, right? So just remember the glomerulus and then the proximal convoluted tubule, descending loop of Henle, ascending loop of Henle, and then the distal tubule, and then the collecting duct. So you just need to remember those main things, right? Just remember those main things as to how the urine is formed. We